Can you see my screen? Hello? Can you see my screen? Please, can you see my screen? Mr. Ali? Hello? Can you see my screen?
Sorry, sir. Yes, I can see your screen, but it's quite small, sir.
No. It's when you're going to. Now, this is the right screen.
Yes, sir.
Is it bigger now?
Yeah, I turned it. I made it horizontal view, so I can see it
Okay.
normally.
Okay.
Thank you, sir.
So, my name is Zaro Leonard. I'm going to be the anchor for this training. So, basically, what we are going to be looking at is we're going to be looking at statistics, then biostatistics. Then we're going to be looking at how to use a particular software, SPSS, at this level. So, are you following?
Yes, sir. I'm here with you, sir.
Okay. So now, we are going to, first and foremost, this class is going to be divided into several classes. The first one is we'll look at introduction to statistics. Then we'll look at how to choose the right statistical test. Then we'll look at how to do normality testing. Then we equally look at then we'll now start taking different life examples on the different aspect of test. Then I'll also give you some work to do as such too. So today we are going to be starting with the introduction to statistics. So now I know at your undergraduate level you must have had to know what statistic is. So when we talk about statistic, basically we are talking about the practice or science of collecting, analyzing, organizing, analyzing numeric data. So what we are saying in essence is the discipline or the study that concerned with collecting, organizing, and analyzing data is actually referred to as statistics. So, and the reason for study of statistics is to make sure that you have you make inference from a population to be able to give an informed decision. Now, what I'm saying in essence is, now, if you want to study the whole population, let's take Nigeria as an example. You want to do a particular study. Let's say you want to check since you are in hematology, let me use hematology example. You want to determine the mean full block, the mean hematology PCV level or the hematocrit level of all the people in Nigeria. You know that is not going to be possible because of the number of population. So what you do is um, you will choose a representative population, and after you have done that, you collect the sample, you analyze your sample, then you now determine the mean. So having determined the mean from that sample, then because the the sample you collected rather, is representative of that population, then you can make an informed decision based on that. So that's that. Then when we talk about statistical and data analysis, we are simply talking about the when we say statistical analysis, we are talking about the means of investigating trends or patterns and relationship using quantitative data. Now, what we are saying in essence is when you want to determine a trend, let's say like the example I gave just now, you determine the hematocrit level concentration or in all the various people in Nigeria, the different category of people in Nigeria. They now want to check the different trend. For example, male hematocrit, how is it? Is it different from female hematocrit? The hematocrit from the north central, is it different from the one from the southeast, from the southwest, from the south, and all of that? So those are what can actually be that you can use. Then another thing you may want to check, does diet that this individual do they influence the hematocrit level. So for you to do all of that, you use statistical method. Now when we talk about data analysis, we are talking about is a process by which you actually inspect, clean, then transform or model data with the goal of discovering useful information. Now, what is the purpose of that useful information? So that you'll be able to make what we refer to as informed consent. So that is very, very essential.
I'm with
So
you,
that
sir.
is um, so that is that. Then another thing we want to look at, I've been mentioning data. Now, when we talk about data is actually the measurement that is obtained in any study. Whatever information or the information you collect or that is collected in any study is referred to as data. Now, the question is, why do we collect data? Now, from what I've said so far, you know that the reason why data is collected is for you to have to be able to get useful information or useful outcome, to get outcomes um, that are essential for you
Meaningful.
to be able to make concrete decision. Now, another thing is the reason why you do statistics general is for you to actually help you as a researcher, or to help your client to be able to what to organize and interpret their data. So the reason for statistics is basically to organize and for you to also interpret those data to have useful uh, information that will enable your client or enable you to make informed decision. Now, when we talk about statistics, we have two broad types of statistics. There's the aspect we refer to as the theoretical statistics. Now, this is the aspect that deal with the mathematics, while we have the applied statistics. Now, the applied statistic is that aspect of statistics that have to do with the application of statistics. Now, under this applied statistic, we also have two levels. We have what we refer to as the descriptive statistic. From the word descriptive, it means is the aspect of statistics that have to do with describing organizing, summarizing, and presenting your data. Why the influential statistic have to do with the aspect that helps you to draw conclusions about a population based on the sample that you have observed in a sample, based on the data you have observed in a sample rather. So what we are saying is um, the descriptive statistic only you just help you to organize and summarize and present your data, whether in tables, in charts, and all of that. But when you want to make decision, want to check the relationships, you want to check the outcomes, you want to check if there are relationship between the different arms of the sample you've collected. You do that by using influential statistics. For example, when you are talking about for example, mean, media, percentage, and all of that, all those are descriptive statistics. But when we are talking about or charts, tables, figures, those are descriptive statistics. But when you are talking about ANOVA and analysis of variance, you are talking about t-test, you are talking about chi-square, you are talking about regression, correlation, and all of that. Those aspects are actually influential statistics because based on the information you are getting from them, you will be able to actually make you'll be able to draw conclusion. For example, if the p-value or the t student test, if the mean between two group of sample, for example, if there is a, let's say we are talking about hemoglobin concentration, the hemoglobin, the mean value of the hemoglobin level of male is different from that of female, with statistical significant differences using T student statistic, for example, you already know that the differences between the mean, the average mean of a male subject or a male sample is different from that of female samples. So that is how what brings about influential statistics. Now, I've already talked about what descriptive statistics, differential statistics. So now, the next thing I want us to talk about is variable. Now, when we talk about variable, we are simply talking about a quantity. We are talking about a characteristic or condition that can change or take on different value. For example, hemoglobin level of an individual can change. For example, if you collect if you take a blood sample from an individual today and you go ahead uh, and determine the hemoglobin level, if that individual after some time probably let's say the diet either improve or they not improve, the hemoglobin level is bound to either increase or decrease. Then another thing too is, for example, gender. You can neither be a male or a female. So those are actually variables. Now, talking about malaria status, for example, you can either be positive or negative. So those are some of the things um, that we are talking about here. Now, the next thing is most times before in most research, before you start your research, you need to determine what your variables are and the relationship between them. So that's actually what is going to determine the kind of statistical test that you're going to apply to that particular study. Now, when we talk about population, we are talking about the entire group of individual. So, for example, everybody in Nigeria make up the population of Nigeria. In a class too, all the pupils or students in that class make up the population in that class. In a school too, all the students in the school made up the population of students in the school. In the hospital, all the patients that visit that hospital made up the population of patients in that hospital. All the doctors, all the nurses, all the medical lab scientists, pharmacists made up all the employees in that particular hospital or in that particular organization, as the case may be. So for you to have a population, meaning that the entire uh, group that made up that particular area is what is referred to as population. So population could be, for example, in the case of Nigeria, the whole population. Then in your school too could also be a population. Your hospital is a population. So that's how what population is. Then, for example, if let's take a student for example. Let's say a researcher want to is interested in actually studying the relationship between let's say the class size of a particular class and also academic performance for population of third grade students. No, note that all the third grade students or pupils made up that population that you want to study. But you know that it's not possible for you to study the whole third grade population in Nigeria. So what you do, you take a representative um, sample. Now, remember that we have different variables here. The first variable here is the class size, but we are trying to check if the relationship between one thing or the other. So the first thing we are trying to check is the class size. Then the second we want to check is the academic performance of the pupils. So that made up the second variable rather. So we are invariably comparing the first variable, which is the class size, compared to the second variable, which is the academic performance. So same also, we may want to also check the relationship between, for example, we may want to check the relationship between, let's say, the hemoglobin level and let's say, and the food, the type of food that an individual, the type of food is one variable. Then hematological parameter is another variable. So another thing too, let's say we want to check the relationship between malaria status and different risk factors. So the various risk factors are variables. Then the malaria status too is also variable. Now, when we talk about sample is usually a part of the population. So what we are saying in essence is, since you cannot study the whole population, all you have to do is for you to now take a sample which is representative of which is actually representative. So you just draw your population, determine your population, then you now determine the sample size. That's why for most study you will always see, they will always ask you what is your sample estimate, because they know you cannot study the whole population. So you have to tell them what your sample estimate is. Now, for you to have a good study, you must make sure that your sample is representative of the entire population. So if your sample is not representative, then you will not be able to answer questions regarding the population. So that is also very important. Now, this is a graphical representation showing the relationship between sample and population. Now, we've already agreed that um, the entire population, all the individuals, of interest are the population. Now, the next thing is the sample is selected from the population. Then the sample that is selected, you collect the data. Then the sample is selected from the population. We've said that already. Then the next thing is the sample, which is the individual selected to participate in the research study. Then the result of the sample are then generalized to the population. So what we are saying in essence is, now if you collect sample, if you have a population, you collect sample from the population and you do a study on that population, then the result from this sample, you can now make inference to this population. So that's why when you do studies, when you, for example, you've collected sample from let's say from married women, or let's use, okay, let's say smokers, for example. You collected a blood sample from smokers, representative sample of smokers. Then you determine the different let's say the different analytes or whatever you determine from the blood sample. Then you cannot make a general statement based on the population. You cannot say that let's say your study was smokers in SCT, for example. Then you collected, let's say, 500 smokers. You collected their blood and you determined their white blood cell level. Now, having determined their white blood cell level, if you want to make your, if you want to conclude, you are not going to be say the blood, the white blood cell of this 500 smokers I collected. No, because your sample size was correct, you can say the white blood cell of smokers in LCT is this. But for you to be able to make that statement, you must make sure that your sample too is well distributed. For example, we have 6 area councils in Abuja. So you can't just collect from AMAC, for example, and generalize it as for the whole FCT. So you have to collect sample from Abaji, you have to collect from Buhari, you have to collect from Kuje, you have to collect from Gwagwalada, then you have to collect from Municipal, that is AMAC and the like. So after you have done that, then you will be, whatever conclusion you are now making, you can actually make it to FCT, because you collected sample from all these from the whole area, not just only a segment. So is that are you following?
Yes, sir, it is clear, sir.
Okay. Now, I made mention of variable when we started. Now, I need you to know that a variable could be qualitative or quantitative. When we say something is qualitative, I know you will have idea here, because when we say we have a qualitative test, quantitative test, and semi quantitative test. So when we say qualitative, we are simply saying something that have to do with present, absent, and the likes. Then when we talk about semi-quantitative, we are talking about something that is ranked. For example, high, low, medium. Then when we talk about quantitative, we are talking about something that is quantified, that have an exact value attached to them. You are no longer talking about high, low, or present, absent. You are talking about the exact amount. For example, let's say you did malaria test, then qualitative is either the person have malaria or does not have malaria. Now, If you want to now talk about semi-quantitative, that's when you are talking about 1 plus, 2 plus, 3 plus, as the case may be. But when you want to talk about quantitative, we are talking about when you are doing parasite density, parasite count. You are determining the exact amount of parasite, malaria or plasmodium parasite that is in that particular blood. So that is what we are talking about. And for this, when we're talking about qualitative, we have two types. We have the nominal, something to nominate. You can either nominate, in this case now, either something is present or absent, yes or no, male or female, present or absent, as the case may be. Then when we talk about ordinal, from the word order, meaning is in rank, is in hierarchy. It's either high, uh, low, medium, and the case may be. But when we talk about quantitative, we are talking about something that is either discrete or continuous. We are going to take that further. Now, still talking about variable type. Now, variable can actually either, you should not be confused. Anytime you hear someone say it's qualitative variable or quantitative variable, and the person is also saying that it's categorical. So once you hear something, when someone say is a categorical variable, is also talking about that is qualitative variable. Then when someone say is quantitative variable, it could also say is a numeric data or numeric variable. So we are going to actually hear a lot of this as we continue. So now the next thing that I also need you to really understand is there's what we call dependent and independent variable. When you say something is independent, it means that it's not dependent on anything. Impact of another thing does not affect it. But when you say dependent, it means that the impact of another thing affects that particular entity. So now, when we say cause and effect, something that can be manipulated is referred to as the cause. That why what the effect is what is measured. Now, what we are saying in essence, what is causing this effect is what is referred to as independent. Why the upon what the effect is fair is referred to as dependent variable. Now, for you to understand this further, there's this description. I don't know if you are a farmer or if you have ever done farming before, but
No,
I
sir.
okay. So I want to use this as an example to illustrate to you so that the message better be um, will better be understood. Now, when we say dependent variable, we are talking about a variable that the researcher can change or a variable that can actually change. Now, in this case, we are looking at fertilizer. And fertilizer, we have different manufacturers. We have we could have what's it called? We have NKP. We have different types of fertilizer, as the case may be. So um, when you apply fertilizer on a particular plant, let's say two fertilizers, you have two groups of fertilizer now. One manufactured by one company A, then another one by one company B. Then you apply these fertilizers to the same type of plant, having the same water source, having the same light source, having the same soil type, and all of that. All the conditions are the same, but the only difference is the fertilizer that you are applying. So you know that nothing can happen to the fertilizer. So this fertilizer now, nothing happened to it. There's no effect on the fertilizer. So because there's no effect on the fertilizer is the independent variable. But you know that the fertilizer is going to have impact on the plant. So because the plant is going to is going to receive the impact or the effect of the fertilizer, then the plant, the various aspects we want to measure for you to be able to determine the different effects of the fertilizer, there are certain parameters you have to take. You have to check the plant growth. How do you check the plant growth? You check the height, you check the number of leaves, then you check the number of fruits that the plant eventually produce. So the effect of the fertilizer is actually checked on the plant by checking the growth of the plant, by checking the number of leaf that the plant is produced. Then the next thing is to also check the number of fruit or the size of fruit, the quality of fruit that the plant produce at the end of the planting season. So the you can see now, looking at this, you can see this fertilizer was applied to two different plants, sorry, the same plant, two different plant of the same group but different fertilizer. You can see that at the end, this plant have more leaves as compared to this particular plant. So it's actually the growth which is measured through number of leaves and the fruit that will also uh, uh, determine. Now, I'm going to ask you a question here to know if you are following. Now, assuming as a researcher, you want to check the risk factors that predispose someone to malaria. Now, you collected people's blood, then you did the malaria parasite, plasmodium, you determine the plasmodium status of the various bloods you collected. Then the next thing, you also collected some question. You also administer some questionnaires, and the questionnaire was you asked them if there was water, stagnant water in where they stay. Is there bush in where they stay? Is there do they sleep under mosquito net and all of that? Now, I want you to note, I've mentioned four variables now: malaria status of the individuals. Then we are checking the stagnant water status of the environment of where those people stay. Then we are checking the whether they sleep under mosquito net or not. Then so check if there is bush in their surrounding. So based on these four parameters that I have named now, which is dependent and which are independent, which is which is the independent and which is dependent.
Um, for the independent variable, I think
Yeah.
that has to be it will include the stagnant water, the whether they stay
Okay.
under mosquito nets, and
Okay.
just basically what are inside the questionnaires. That would be
Okay.
the independent variable. Why the result would be the
Yeah.
dependent variable, sir.
Great. Great. That's good. All right. So let me just check if there's anybody joining so that I'll be sure we are not leaving anybody out. Okay. So, all right. So let's continue. So we are going to move further. Now, I make mention of nominal. I said a nominal variable is actually a qualitative variable that categorize or describe or name an element of a population. So I want you to hold that very, very important. So anything that just categorize, describe, or name an element of a population is a nominal variable. For example, you are a male, you are a female. You are either a male or a female. You either have malaria or you do not have. You either sleep under mosquito net or you do not sleep. is neither there's stagnant water in your environment or there's no stagnant water. So those are actually categorization, description, name. So those are nominal. Then when we talk about ordinal, it's also a qualitative variable that incorporates um, an order position or ranking. So what we are saying in essence is when you are not talking about ranked, talking about order, talking about ordered position for like I described earlier, after you know, okay, you already know there's plasmodium. It's just like when you use this rapid malaria kit, it will tell you there is plasmodium or no plasmodium. It will not be able to tell you the quantity or tell you the intensity. So if you want to determine the intensity, you have to do um, uh, first and foremost do a quantitation in to some extent by doing uh, either plus minus plus 1 plus 2 plus or 3 pluses. So because you are actually putting all these things in a rank position, then that is nominal. Also, when you are talking about, okay, let's say you do glucose level, is either it is normal, it is low, or it is high. So in that category, in that regard, you are talking about ordinal. Then when we talk about discrete variable, discrete variable is a quantitative variable that can assume a countable number of variable, a countable number. So what we are saying invariably is that a discrete variable can assume a value corresponding to isolated point along a line interval. So that is, there is a gap between any 2 points. Now, between 1 to 2, there is a gap. 2 to 4, there is a gap. 4 to 6, there is a gap, and so on. Then when we talk about continuous variable, we are talking is also a quantitative variable that can assume an uncountable number of variables. So what we are saying in essence is you cannot really determine uh, you cannot really a continuous variable. You can also assume a value along a point line, but in this case, every possible point between any two value. So what we are saying is a continuous variable can be at any point, but for a discrete variable, it has to have a gap. Then you can actually determine what is the difference between the two. For example, the difference between 1 and 2 is 1. So just like that. Now, okay, we've talked about this. Then we also have what we refer to as measurement scales. So you can neither measure your variable in a nominal scale, in ordinal scale, or or an interval scale or ratio scale as the case may be. So any questions so far?
No, sir. Maybe
Okay,
about the slide we are on at the moment. That's just it. No question, sir.
okay, okay. All right. All right. So let's continue. Now we are at a very important aspect, which is the first problem that as a statistician that you are going to face is knowing when to how to collect your data. Because if you do not collect your data well, the result you are going to get will not make sense. So the first problem a statistician will face how to obtain data. Now, the reason why it's important is it is also important to obtain good and representative data. What we are saying in essence is, yes, having determined what data you want to collect, you must make sure that the data you are collecting is good and the data you are collecting is representative. So what I'm saying in essence is, for example, you want to collect, just like the example I gave earlier, you want to determine the hematological parameter of smokers. But having because you are not seeing enough smokers, you now decide to collect the sample from anybody at all, anybody that make itself available. Yes, you may collect representative data. You may collect a data that is actually quite enough, but the question is the data good? Is it representative of the uh, population you intend to study? So you must make sure that the people you're going to collect this data from, that they are the actual population that you are supposed to be dealing with. So, and if you are not collecting the right data, you will not be able to make a good decision. Now, there are different sampling techniques. We have what we refer to as probability sampling method and non-probability sampling method, or probable sampling method and non-probable sampling method. Now, when we say probable sampling method, we are talking about the sampling technique that is based on probability. Now, why the non-probable sampling method are those methods that do not depend on um, probability. So, in essence, probability method are scientific based. When I mean scientific based, is that they are methods that are designed, why not probability are methods that are not designed. One of such is simple random sampling. Now, under simple random sampling, I want us to go to this slide. Now, this is an example of probability, probable sampling method. You can see this is simple random selection. Yes, we have the same number of individuals here, but random in the sense that you take the first one, then you leave two, you take another one, then you leave three, you take another one, then you come down, you take one again, then you leave one, then you take another one, then you leave the remaining. This is random. It is random. It's not, you just choose based on you feel. Then the next one is what we refer to as stratified. From the word strata, meaning that you are grouped into different groups. Now you can see here, now all the people that are on green are in one, are grouped together, while those on blue are grouped together. So stratified, this is stratified now based on the color. Now you see that, you see that here it takes the first one here, then it moves down to this one and take the middle one. Then when it comes to the blue, it takes the second one, then it leave three, then it move here and take the next one, two. So the systematic sampling is actually a very robust and scientific method of collecting data rather. Now you notice that here they are grouped. He leave the first one, collected, then after two people collected another one, after another two people collected another one, after another two people collected another one, after another two people collected another one. So you can see that this is systematic. Then we have cluster. Now, cluster in the sense that these people are clustered into different, okay, you have to, they are clustered into three groups. So you see that we leave the first one, we take one cluster. Then we leave this other one, we take another cluster. So I don't know if you are following that.
Yes, I am, sir.
Okay. So now for the not probable
Sample
examples,
method.
yes. So you can see that there's what we call convenience method. Now he just collected all the one that are just close to him. Then the next one is voluntary response sample. Now he just call for response, then it's those that responded that he sample. Then we have purposive technique. In this case, you just look for anyone that look important to you, then you collect it. Then the show ball is like you also stay at the middle, then you collected anyone that are just that your hand can easily get to. So how do you actually collect your sample? Now, one of the way through way you collect your sample is through experiment, which is very common with us. For example, all your laboratory techniques are based on experiment. You collect someone's sample, then you determine the hemoglobin level and all of that. Then another that's one type of experiment. But in scientific research, there's the experiment that is very, which is the one you introduce intervention. For example, let's say you have, you always have what we call case control kind of study. Now you have a group of people or group of laboratory animals. Let's say you expose some to sun, you expose the other ones, you not expose them to sun, or basically you gave some one type of food, then the other ones were not given the food. Then after a time, you collect their blood sample and determine some certain analyte on the sample. So that is one experiment. So um, in this case, the investigator control or modify the environment and observe the effect on the variable under study. Just like what I said, you the same laboratory animal, you do a baseline test to be sure that all of them are the same. They have the same hematological level. They have the same they are the same condition. Then after that, you now separate them into two groups. Either you change their environment, or the next thing you will change their diets. So that is one way of introducing intervention. Or another one, you train them over on certain something. Then after, you now observe the effect or the outcome as the case may be. Then another way of doing or collecting data, method of collecting data is through survey. Now, survey is usually data are usually obtained by sampling some of the population of interest. So what we are saying in this case, the investigator does not modify the environment. Just like, for example, you come collected sample from all the people that are in a particular area. You are not changing anything. You are just trying to check the state of that particular analyte. You are not introducing any form of intervention. So in this case, it is a survey. Then the next one is census. Census is like 100% survey. So what we are saying is that you do census, you collect sample from all available population, all possible population in in an area. Let's say all the people in FCT, you collect all their sample. So that is census, for example. So that is that. So the introduction here today will this is where we'll draw the curtain. Then under the statistics, we have statistics and technology. before the before the inception of technology, people have been doing statistics. Remember, you have to write a whole lot of formula to do chi-square, for example. But in the era of computer and in the era of technology, you see that these have actually made things easy. You just press a button, then all your results will come out. So there are actually a whole lot of statistical packages that are available. But the one that usually you always learn first is the SPSS and the Excel. But in this training, we are going to be focusing on SPSS. So Excel is like the baseline for everyone. Then after you have learned SPSS, then you can decide to take the training further by learning either Arrow, learning Python, learning Stata, SAS, and several other software that are available. So I will stop here and take your question if there's any. So I will stop sharing now. Then I will take your question uh if there's any question at this moment.
There's absolutely no question, sir. It was
Okay.
beautifully explained, sir. Thank you very much. I really appreciate,
So
sir.
we'll meet
I will
tomorrow
have to
same
go
time.
through it. Yes, sir.
So I will also share on the group the software.
And
Then
the
you just
slide, the
yes,
recording.
of course. I will share the
All right.
is actually I'm actually on YouTube. So I will send the YouTube link so that you can view it on the YouTube.
Okay. Okay.
Yes.
All right, sir.
Yeah.
Thank you very
Thank you.
much.
Good
Have a
night.
wonderful
Yes.
night, sir. You too, sir.
Yes.
Bye, sir.